So basically, we're a developmental neurobiology lab, always been interested in how the brain forms. And we've done work on sort of those two major areas, namely the differentiation of neurons and their specification is different from one another, and then aspects of axonal growth in order to wire up neural circuits and generate this mess known as the brain. And in doing all of this work, you know, at some point you figure it out, right? I mean, conceptually, it stops being terribly challenging. You understand how to at least to go after a certain problem. And at some point, you're confronted with the inevitable in biology, which is variability. And of course, you try to control that. And in flies, you can do that quite well. And you can't seem to get rid of that problem. And at some point, I decided that, you know, this is actually what's interesting. Not how global pattern forms, but why, despite controlling as many elements of the process, the genome, the environment, et cetera, as you can, you really cannot change the fact that this is variable. And so where does that variability come from? And that's the story I want to share with you today. And I guess like everybody in the room pretty much, you know, as soon as you start saying that, well, I don't want to look at the mean anymore, I'm interested in the error bar, all your colleagues start saying, you know, why are you wasting your career? You're doing well, just stay focused, et cetera. So we've been a historically unfocused lab, and that's the way it's always going to be. And what I want to share with you is our attempts to make some inroad into this question in not what generates the brain, but what generates the I brain, right? Singular brain of one individual. Now, what makes us different? There are at least 3 different sources, and they're two of them are fairly obvious and heavily studied. You've heard from Chris Walsh, for example, about genetic differences in the population that predispose or even can cause different individuals to become behaviorally different, so different, in fact, as to be classified as having a disorder. We have all experiential differences that contribute to our personality, and those are not necessarily on the scale of, you know, rapid plasticity. But I'm sure everybody in this room had had some kind of a super pleasurable or super traumatic experience that then left an indelible mark on you for the rest of your life or for a very long time. I made the mistake of taking my 3-year-old son, who was insisting to go to McDonald's at some point because he wanted to play. He ordered the chicken McNuggets. Of course, he didn't finish them. So I sat there watching him jump around and eating one of those chicken McNuggets. I got sick, not surprisingly. I actually could not smell or eat chicken for the next 10 years, honest. Right? So that was clearly, you know, something that was an experience-dependent personality trait, at least for long periods of time. And these things are reasonably well studied by a lot of people, but there's a third source of variation that people who are developmental biologists as opposed to neuroscientists by training actually know quite a bit about, but that neuroscientists have neglected for a long time, which is that there is intrinsic developmental variation. And developmental biologists call that stochastic developmental noise, for lack of a better understanding, which means random stuff happens during development. There's nothing you can do about it, and that contributes to differences in animals. And the question is, does this actually happen during the wiring of the brain, and does it actually matter? And so that's the challenge. The idea is that genetically identical brains nonetheless suffer developmental noise and molecular noise during development. That creates slightly different neural networks, and those slightly different neural networks actually contribute to the personality of an individual. And the idea is to use then fruit flies, which are sufficiently simple and sufficiently complex in a way to study that problem. And again, like everybody in this room has sort of a similar story, which is you say, well, let me send this to a funding agency, right, and see if it can get funded. And I was driving Tom home when he visited our institute and explained to him this idea. And he said, that just sounds great. Can you write it down in an email and send it to me? And the rest, as you know, is history. I'm here. And, you know, got funded. So that's great. So how do we tackle this problem? Well, to tackle this problem, you need a neural circuit that you know something about the anatomy, and you can show is highly variable. And in this case, this is a cluster of neurons that I had the fortune of stumbling upon when I was a postdoc. So I discovered these cells and very creatively named them the dorsal cluster neurons instead of the Hassan neurons, for example. You know, that's a missed opportunity. But so it turns out that these bilaterally symmetrical nucleus of cells develops from a single embryonic stem cell through a number of divisions, and yet creates about 30% variability in the number of cells. These cells then make a decision to target two different target areas in the visual system, a distal target and a proximal target. The names don't really matter. And they do so, again, with a certain level of variation. So they send about, regardless of the number of neurons you start with, you send about, well, really 10 to 14 axons to the distal target, and the remaining set of targets are in the proximal target, right? And so this is an ideal system to understand how variability might be controlled. This is almost never the same between individuals, but furthermore, no aspect of this pattern is the same on the left and the right side of the same individual. So clearly an epigenetic phenomenon. And now you can begin to ask, might this contribute to individuality, and how? So how does this pattern arise? This is all published. So I'll just give you the upshot. This was work by a former PhD student, Marion Langen, in the lab. And what Marion suggested was that the pattern emerges not as a result of every neuron knowing what it's going to do, but rather as small subsets of neurons having conversations in a competitive way, whereby one of every 5 or 6 neurons is allowed to keep an axon in the distal target in a topographic map, and sending a signal to all the others to go back to the proximal target. Okay? And so she was able to demonstrate that this is true, and I'm just going to show you an example of this. And so basic idea is here. All the axons initially would go to the distal target, and that is true developmentally. Then they would have a conversation. These 5 or 6 cells would decide among themselves who gets to keep the axon here. And in this case, it's, let's say, the red cell. Now, and this is a completely stochastic decision. And then the red cell would tell all the other cells, go back. And you would then have to go a few cell diameters away to a cell that's no longer in touch with this winner in order to have a second winner, and so on. If this is true, you can mutate one of those cells. You can load the dice, make that cell win the competition. And we knew that this was driven by notch activity, so you know, a signaling pathway, which mediates something called lateral inhibition or mutual inhibition. You could mutate the notch protein in this particular cell, and you could rig the competition. So now the orange cell will win the competition. But if you move 5 or 6 cells away, you're going to get one winner, and then you're going to get one, you know, the same exact pattern once again. So you can change the decision of one cell, but that will not affect the overall pattern. The other prediction, on the other hand, is if you now mutate multiple cells that are neighbors of one another, you sort of destroy their ability to communicate, and in that case, you will have multiple axons, regardless of their mutant or wild-type status, attempting to target the same area. And so we tested this prediction for mutual inhibition. I'm going to show you just this, what I think of as the clinching experiment from a whole series of data, which is the following. So these are stochastically mutant cells for this particular protein called Notch, in which all the cells are labeled in red, and only the mutant cells are labeled in green. Okay? And so you can now get sort of multiple cells, either isolated or together. And as you can see, when the cells are isolated, it is the mutant axon that will be in the distal area, but it will be alone. There will be some distance, and the next cell, which happens in this case to also be a mutant cell, will have its axon. From here, you will have one of the wild-type cells. You get a beautifully normal pattern, even though you've decided which cell is going to actually innervate. Whereas in the cluster where you happen by chance to create multiple mutant cells right next to each other, you can see that multiple axons are attempting to innervate the same target. So you can demonstrate quite clearly that this is a stochastic process that results in a stable pattern, even though no one particular cell is predestined to do anything in particular. And this, of course, allows you to generate stereotypy in the pattern, but individual variability among different neurons, or among the left and the right side of the same neuron. So asymmetry within an animal, variability in the population. Marlene Schlieder-Cech then went on, as another PhD student, went on to show that not only is the axonal targeting process stochastic, but also this beautiful, highly stereotyped pattern formation actually comes from a completely stochastic redistribution of a limiting amount of a molecule called the EGF receptor among early developing branches. So initially, these axons actually make a ton of branches. And there's a limiting amount of this molecule that's stochastically recycling. And every time it enters one of the branches, it collapses. And so, of course, the process will keep repeating itself until there's one branch left, and that's it, right? One branch left on either side, and you get this beautifully stereotyped pattern from an initially completely noisy process, right? So what this tells us is that wiring specificity is not a problem of address codes. It's not a problem of every neuron necessarily knowing where exactly it has to go and who exactly it has to synapse. It's a pattern formation problem. And if you accept that, then in fact variation inevitable noise in development in molecular processes are actually used as part of these developmental rules that you heard a little bit about from Mike, to generate variation in neural circuit wiring across genetically similar individuals. But if you know the rules, you can, of course, now transform using these rules to create new anatomies, not to create just massive disruption, but actually subtle changes in the anatomy of the brain and ask, does this matter developmentally? And if you wanted to do that, you would need to know something about this neural circuit, and you would need to first demonstrate that individual flies have personality traits, that they actually have behaviors that are unique to them, that are stable over time, but different between individuals. And so this is what we first set out to do, and this is the work of a postdoc in the lab, Gerrit Linneweber, and so this is now all unpublished new data. And basically what he was able to look at is this dual stripe fixation assay. So flies love to fixate on black stripes on white backgrounds. Nobody knows why, but they love high contrast, and they will do that. And if you look very carefully, the fly here is attempting to reach the stripe. It can't because we've put water, and then it will go back, and it will basically go back and forth between the two stripes. And about 50% of the population goes in a relatively straight lines back and forth between these two stripes. And if you repeat this experiment with one individual multiple times, you can create a heat map for that particular individual over time and, sort of establish its own behavioral parameters. And now what you can do is ask, is this particular behavior stable in this one individual? So this is just a snapshot of a lot of data. So basically, what we do is we test the same fly over 3 weeks. So we do an experiment. We take the fly home. It rests for a day. We repeat the experiment, and we keep doing that for weeks. And I'll just show you a bit of the data here. So these are 3 different individuals. And this is a fly that sort of leans to the right every time it wants to walk between the two stripes, and it will do that on day 1, it will do that on day 3, and it will do that 3 weeks later. And this is a fly that loves to meander in no particular path before going to the two targets. This is a fly that sort of mostly walks up and down in the center, but has a very wide distribution to its path, and so on and so forth. And you can, again, create these heat maps that are summaries of the behavior of one individual, and then you can ask about the correlations, and you get coefficients of correlation of 0.8. This is a highly stable behavior in one individual, and almost no two individuals are the same. Okay? So flies have personalities when it comes to at least reflexive attention behaviors. What happens if you silence this circuit that we had, I'm not showing you the data here, that we know is involved in stripe fixation, and these neurons actually fire in response to flies being shown fixed objects? So if we silence the activity of this circuit, which remember is intrinsically variable, all the flies, or almost all the flies, upward of 90% of the flies, become hyperfixators. They straddle a very narrow path, up and down, back and forth, and sort of you wipe out the individual variability between different individuals in this particular assay by simply silencing an intrinsically variable neural circuit. So flies have personalities, at least when it comes to reflexive attention, and we can eliminate their individual tendencies by silencing an intrinsically variable neural circuit. That does not, however, show that the variations in the anatomy of this particular circuit underlie the variability in the behaviors of individual flies. And that's what I'm going to just show you very preliminary data for. So basically what Gerrit does is he does the experiment. He takes every individual. He does, now that we know that the behavior is stable, we only have to do it over 3 days. And so basically he creates a behavioral map, a heat map for one particular individual. He dissects that individual and asks how many neurons are there on the left, how many on the right, how many axons in the distal target, how many axons in the proximal target. So he extracts the anatomy of that one particular individual. And he starts to draw correlation maps. And there are very important correlations, and they're quite specific, and they're not all over the place. And let me just show you a few. This is grayed out because it's the exact mirror image of what's up here. So this is left-right asymmetry. This is right-left asymmetry. So basically, what we find, for example, is that the asymmetry in the innervation of the distal target correlates with a number of behavioral parameters only when the fly is in motion, but not when the fly is static. On the other hand, the asymmetry in the innervation of the proximal target, which is much larger, correlates with what happens when the fly, for example, the angle and axis deviation, which are deviations from the straight and narrow, correlate with what the fly is doing while it's stable, while it's static. So the flies often stop in the middle of a behavior and orient themselves. They can stop up to 10, 20 seconds and then decide to walk again. And when you now dissect away the behavior in motion versus the behavior when the fly is stationary, you will find different correlations with the anatomy of this circuit. So I think there's pretty good evidence that, this is what I've just told you. And so here's the to-do list. Of course, what we are in the process of doing right now, this is an example here, is to create new circuit anatomies and query the behavior. We already have some evidence that we can actually alter the behavior of a given individual stably by changing the anatomy of the circuit. We want to look at the emergence of this variation live. So we've now developed, together with Robin Hiesinger's lab, ways where we can image the development of the entire process of brain development at single filopodia resolution while looking at the entire brain. And so we want to look at the emergence of variation during development under normal and mutant conditions. We want to correlate neural circuit anatomy with neural activity using calcium imaging, so to ask whether when you generated a slightly different anatomy, do you generate slightly different patterns of activation of these neurons. We would love to look at on the social effects of individual behavior. And this has been a major challenge because the faithful tracking without losing identity of a large number of organisms has been very, very difficult, particularly in flies. And we have just solved this problem very recently. And this is just an example of showing you the tracking of about 30 flies or so, and over an hour, and we don't lose identity of a single individual. We'd like to understand the relevance of this non-genetic diversity in the brain to the population. And we can discuss about that. So, is this of any evolutionary advantage? And finally, I don't know, 500 years down the line, is to be able, and this came out of a conversation with Christopher Bouchard at the Lawrence Berkeley lab in California. The idea is, can we make wireless nanoparticles that are targeted to certain proteins small enough that we can actually feed them to flies during development and create animals that have specific neurons with wireless control nanodevices, and we can start to wirelessly play around with the behavior of an individual and have some fun. I actually suggested this to the Microelectronics Center in Leuven, where my lab was until this year, and they told me to go home and stay a biologist. So, but the Lawrence Berkeley people said, hey, that's something we should think about, so that's great. So I'll stop here. I mentioned the people who have done the work. They've been helped, of course, by other postdocs in the lab, my wonderful technicians in the previous lab in Leuven, a lot of funding, collaborations with other people, and I specifically want to highlight the collaboration with Robin Hiesinger's lab, which has been absolutely amazing, and the current funding in Paris from the ICM, various institutes, and of course the Frontiers Group for believing in the idea and funding it. Thank you.
We have time for one question.
Bassem, in the earlier part of your talk with the stochastic crosstalk between neurons going to two targets, would the solution be different, or the conception, if it wasn't the neurons talking, but if the operational unit was the growth cone or the axon?
Yeah, that's...
Could it be a tiling out there and the
Yeah.
neurons just a supply depot?
So that could be in principle, but I think the evidence, I mean, we have very strong evidence that this is actually the classic lateral inhibition notch nuclear pathway, and that it's happening at the level of the cell body and then being sent through the modulation. So you turn a stochastic process into a binary switch, whereby the cells that have low notch nuclear activity now have higher levels of J and K signaling, which simply drives axonal elongation. So basically the idea is now those low notch cells basically stabilize their microtubular structure to a point until synapse formation can occur, and the other axons sort of, you know, lose their microtubular stability and start retracting back to the lobula. And this we actually can see happening live. So it's pretty clear to me that it's happening at the level of the neurons and not at the level of the growth cone.